And Dylan, get better. Heal up. She'll be at the reception. Oh, great. Okay, good. So our next speaker is Diana Elizabeth Maciel. She's at the University of Washington, a rising junior, and she's in the Jayadev laboratory. And your mentors are here. Your mentor is here, right there. No, I know. Come on up.
Good morning, everyone. My name is Diana, and I spent my summer here at the Jayadev lab. And my talk will be on the response of PSEN2 N141I microglia to 7-day amyloid beta exposure. So while majority of Alzheimer's disease cases are sporadic, meaning late onset and non-inherited, a portion are familial, and those are inherited and often early onset. So familial Alzheimer's disease is the interest for my project, and I will focus on PSEN2, one of the 3 known genes whose variants cause familial Alzheimer's disease. It's been recognized that the N141I mutation found in PSEN2 is associated with disease-causing behavior and is of interest in familial Alzheimer's disease studies. But regardless of form, both familial and sporadic Alzheimer's disease have these pathological hallmarks: the buildups of amyloid beta plaques in the brain and tau neurofibrillary tangles, both of which affect neuron structures and communication between cells. In addition to these hallmarks, increased immune system inflammation and activation of microglia in the brain have been observed in Alzheimer's disease. And microglia are the brain's innate immune cells. And the cause of microglial inflammation is still unclear, but one possible hypothesis is that it's from amyloid beta itself. So to understand this better, we have the 2 following questions. How does 7-day amyloid beta treatment affect microglia in a human genetic model of early onset familial Alzheimer's disease? And how does a PSEN2 mutation affect microglia response to amyloid beta exposure? In our lab, we use iPSCs to model Alzheimer's disease, and these are cells reprogrammed to be used in disease modeling. The iPSCs are then differentiated into neurons and microglia. And in the case of my experiment, I looked at cell populations where neurons and microglia were grown together in a co-culture. And so we had 2 co-cultures, both of which had the wild type or healthy neurons. And then they either had the wild type or microglia or the N141I mutated microglia. And co-cultures underwent 1 of 2 treatments, either untreated and fed normally or given amyloid beta for a 7-day period. And our goal then was to measure PSEN2 expression and observe morphology. And so the reason why we observe morphology is important because microglia is known to change in Alzheimer's patients from this healthy homeostatic and branchy state to a more amoeboid state right here. And so to do this experiment, we used immunocytochemistry. And so ICC involves the use of primary and secondary protein antibody staining, or primary and secondary antibody staining to see desired proteins. And I used 3 antibodies. IVA1 which shows microglia, MAP2 to show neurons, and PSEN2 for PSEN2. And so once done, we mounted our cells onto slides, and they look like this. This is one of them. We had a total of 4 slides based on our 2 genotypes and our 2 treatments. And for imaging, we used an SP8 confocal microscope at objective 40x with oil. And we ended up taking 5 images of each slide in different locations. So here are photos that we took for the wild-type microglia untreated, the wild-type microglia with 7-day amyloid beta treatment, the mutated microglia with untreated, and the mutated microglia with 7-day amyloid beta treatment. And so visually, what stood out to me was that our cells, especially for the wild-type microglia here, they seem to maybe increase in size. And so I wondered if those changes could be significant. And while my pictures may have suggested so, the visual observations aren't enough to come to a conclusion. So we had to find a way to quantify our expression and size. And that's where Imaris analysis comes in. So Imaris is a software that allows us to compile individual layers of microscope images and create the 3D measurements of cells. And so for here, we took our nuclei, which are seen in dark blue, and our microglia in the bright green, and made them 3D shapes that could give us volume and signal intensity statistics. And so for our first results, focusing on the volume of microglia surfaces, the first thing we found was that it's for our wild type microglia, there did seem to be a change in volume after the amyloid beta treatment. So here on the green shades, that's the wild type microglia. But we did not find a significant change in our mutant microglia. So there was no significant volume change. And then for our other results, we looked at the intensity of PSEN2. And so if we recall, PSEN2 is important because the presenilin protein increases with immune cell activation. And so our first conclusion from this was that the mutated microglia seen in the blue shades did have a higher PSEN protein expression in it compared to the wild type microglia in pink. And that was just baseline regardless of treatment. And then the other thing we found was that the 7-day amyloid beta treatment did increase PSEN2 proteins in both genotypes. And so for our takeaways in terms of morphology, 7-day amyloid beta treatment does seem to alter microglia in some way, specifically for here, volume. And in terms of PSEN2 signal, PSEN2 expression does seem to be higher in our mutated microglia than the wild type at baseline. And our 7-day amyloid beta treatment also increases PSEN2 expression in both genotypes of microglia, but seems to have a higher effect on the mutated microglia. And there are other adjustments we can do to this experiment. I think the biggest one being increasing multiple slides. We only had one slide and just took it at different locations. But we could also do other treatments such as p-tau, and then ultimately we can also mark for additional cell types such as astrocytes. And in the far future, because we suspect that mutant microglia are more aggressive and activated, studying them can help us understand if they do worsen familial Alzheimer's disease, and that can later help us design immune therapies for familial Alzheimer's disease. So lastly, I want to thank the NSSSP admin for giving me this opportunity to be here today, and as well as the Jayadev lab for their support this summer, specifically Sumi and Akena for helping me put this project together. Thank you.